So Heidi Dungy is going to talk to us about hopefully convincing us how to change how we do things in forestry, talking to us about technologies of eucalypts and how to use that in Eucalyptus nitens breeding in New Zealand. Thank you.
Thank you, Sandra. Awesome. So I guess the first thing I learned about my talk this morning is that my title was way too long. So apologies for those who can't focus yet this morning and haven't had enough coffee. So really today I just wanted to go through a little bit of New Zealand context, because now I've lived there for 18 years, I know a little bit about it, and some genomics technologies, what we've been playing with in the nitens populations, and then some of the remote sensing work we've been doing, and just introducing some of that concept that although we've done it in radiata pine, how we might want to apply it in the future, also possibly to eucalypts. So let's start with some New Zealand stuff. And there are 2 islands, and it's quite interesting in that most of the weather comes from the southeast, as in like similar to Tasmania, and we have is that it? Yep. So there's basically mountains all the way through. It's quite volcanic and new soil. So the best thing about New Zealand for foresters is it usually gets a lot of rain and it usually has really good soils. So hence we get pretty good productivity. A little bit of history. So one thing I find fascinating is that there were no humans in New Zealand about 2,000 years ago. So Polynesians came round, but there's a bit of debate about that, I guess, maybe around 1,000 years ago, and after that, Europeans about 250, 200 years ago, and so we've had successive natural forest deforestation. Now, so we went from 82% to 24%. And that's actually one of the reasons that they have a lot of exotic forests, that they decided they're going to run out of indigenous forests. And so they decided, let's test and check out what might grow faster and more easily. So they settled on radiata pine after some species testing. So we have forests on around 7% of the landmass. I think that's actually gone down to 1.7 million hectares. And it's unfortunately eucalypts, which I also have a passion for, is only about 1% of that. So just putting that in context, I'm talking about a small part of what we do, I guess. And we've got about 23,000 hectares and around between, I think it's about 13,000 hectares at the moment of Eucalyptus nitens, and it's mostly in the south of the South Island. We have a competition from land use, not only dairy, but hobbits. And Hobbiton is expanding. And that tourism sector, just based on those films, is quite enormous. But let's talk about genomics. Why bother? And in this context, I'm just going to give you an idea of our little breeding population of Eucalyptus nitens. Our eucalypt breeding populations are, let me say, run on the smell of an oily rag a little bit, and then we're allowed to put some research on top of that. So we usually run them as open pollinated breeding populations with forward selection. So really simple, and everything we select in the field trials we then graft up, and or we can take actually straight from the field trial as well. So we really had basically nearly 4 generations established from natural stands and from imports. And I know that I'm talking to a few guys from the SGBA about maybe doing a joint analysis of some of this material. But the end result is we have 2 kind of seed orchards at the moment, which are separate and kind of like the old super lines. So there's no known relatedness between those at the moment, and just keep that in mind. So we've had a little play with the Eucalypt snip chip, and one of the reasons we're playing with it is, of course, we're running open pollinated, and we've up to 4th generation, we're starting to get really worried about inbreeding. So basically, the trial that we're basing this research on is around 4,000 individuals, and we had 691 individuals representing 72 families were put into this genotyping. So why would you bother with genomics? And this is one of the things we get a bit obsessed about, is quality. How do you know that tree's the same tree all the way through the pathway? Because we know we make mistakes, and unfortunately you find a few more mistakes than you'd like to admit to. But you know, when you have your progeny trial, you select it and you find the best tree. It's really good to have a DNA fingerprint so that you can make sure going forward that if you use it as a parent, is the same parent going forward. And we've done a look at, in one of those trials, at parents and sneakiness, and so this is some of our forward selections in that trial, and we basically just did, okay, these are our selections. What is the parental contribution of what goes into those selections? You know, because we don't know the pollen parent. And what we found is basically some of them had a bit more contributions than other pollen parents, and this enabled us to trim out too much of that going forward into the next generation and into the seed orchards, so that we could keep a little bit more control on inbreeding. And we did that with the Eucalypt chip, which at that time was extremely expensive, and we're very, very grateful that it's going to go down to something around $20. And oh yes, I wanted to point out Yaroslav. Yaroslav's been involved in doing a lot of this research as well. And we recently also had a look at rescaling the G matrix, which is looking at using markers instead of the pedigree for our analyses. And what we found, but in this open pollinated population, which is not unexpected, I guess, that basically using the markers had a really profound effect on our genetic parameters, and in particular with stem diameter, which we found a bit of inbreeding depression in our populations. The most exciting thing, though, is generation time, and I've just put this up because this is radiata pine. We haven't done the exercise for eucalypts. But basically we can move, we think, from 17 years to 9 years. Now if you do a net present value estimate on that, it's actually quite a lot of value in bringing that down. For eucalypts, because you still, we put cloning into this example for radiata. Eucalypts, if you can't clone it, it's not quite as good, but it's still a significant improvement, and it really does add value to your breeding program to get that stuff out sooner. Early selection, of course, and we all know that when we look at some of our wood traits, they are quite difficult to measure and very expensive. So we've done the same 600 trees, and we did some very extensive phenotyping looking at wood properties. In particular, if you look through internal checking, which is, I guess you guys sometimes call collapse, density, growth, strain, shrinkage, and collapse as well. So individual blocks were then measured for each individual tree, and all the data was collated. But some of the key take-home messages really were that we did BLUP based on pedigree and BLUP based on, or G-BLUP really, based on markers. And basically, if you look at the BLUP heritability here, this one, and the BLUP accuracy, the G-BLUP here, the accuracy goes up significantly, and the heritability also goes up a bit. But the interesting thing was that there are a couple of things here like tangential shrinkage and stem straightness, and we know that stem straightness, we use our phenotyping, is actually a subjective score of 1 to 9. So that to me tells me that we need to up our phenotyping on that, not that the G-BLUP's wrong. In addition, tangential shrinkage can be notorious to measure and a real pain. So I think that there's some key messages in these results anyway that we can follow up on. And if you look at just the prediction accuracy for the different traits, you can see again that message coming through for the stem straightness, but interestingly for the diameter and height, it actually, although the heritability is low, that still get reasonable prediction accuracy. And if you think back to that diagram, there were 2 seed orchards, right? So Yaroslav did some analysis of different linkage disequilibrium from the different orchards. And clearly we have different patterns from the different orchards. And it's something that I don't really care about, but I want to put them together. So what's the impact of that? I really want to make sure that we can account for these differences in a way that's going to be useful for breeding. So the take-home messages is that basically wherever we're looking in our breeding programs, different selection histories will probably result in different LD decay, such as this, and we have to work out how we're going to take care of that. And there's some good hints in some of the other talks, I think, about haplotypes and segregation that might lead us in that direction. And if we just look at some of the spectral decomposition of the markers, again, some of the populations come out. So here are the 2 different orchards. So we can see clearly a kind of a bit of a difference between the 2 orchards. And there's some Australian Tree Seed Centre in the middle for those Aussies who are interested in that. So that's where our population's at. And so these things are, although genomics is expensive to start with, these are the sorts of things that you get on top of the normal day-to-day breeding information that I think is really useful in how we manage our populations. Okay, so I'm just going to skip into some phenotyping work because I talked about straightness, but I think with some of the new work that's coming on, we can really start to reimagine what phenotyping looks like. So I'm going to talk about a concept that we've come up with at Scion about phenotyping the forest. We have, and really lucky to have access to a large amount of LiDAR data. We also have GIS climate surfaces, digital terrain maps where we can extract a whole lot of information. And we also have a lot of genetic information, not necessarily genotype, but genetic information across the forest. And we put this together in a kind of conceptual feedback loop, which is using this data to try and track the genetic performance across the forest. And this, because we know the genetic seed lots that go out in the forest, will help us understand and track where our genetics is performing well and where it isn't, and also create a feedback loop into the breeding program. Even recognising the worst performers in your forest using this method can significantly increase the productivity. So just show you an example of a couple of forests. Really we've done this across Kaingaroa, we're lucky enough to work with those guys, and Rotorua, which is Lake Taupo Forest Trust. And we take surfaces like this, and you can obviously extract a height surface, so you can model a stand level height surface across the forest, and the digital terrain map. And then you add in things like aspect, wind, slope and landforms, as well as climate. As I said, we've got the GIS modelled surfaces across New Zealand, and additional information from the forester. So we have good forest management information of soils, silviculture and disease. And this is an interesting little exercise where we tracked, we killed these trees using Roundup basically, and tracked their death using hyperspectral imagery in a UAV. So, and as I was saying, although we don't know the genomics necessarily of the seed lots and the structure of the forest, much of the coops across the forest do have genetic information based on their GF plus seed lot. So we've done some analysis using that, and each of these has a trait rating such as growth, straightness and branching, but often it will have something like a Dothostroma resistance rating as well. And within that there are the known parents So at the moment we've done some analyses using the GF Plus seed lot, and we're starting to dig now into the parental information. So some of our early results really are showing that this is the site index, which essentially is the mean top height surface across the forest. Some of this is the seed lot code, which is really the GF Plus number, and basically it's showing a relatively high influence compared with the other things across the forest, which means genetics in that it is a really operational tree farm, right? So it's not normal, it's not natural, but basically genetics is a really key driver to our productivity. And we're also digging down, and Dave Pont and Jonathan Dash at Scion have been really doing a lot of work, and I know a lot of you have seen this slide before, apologies Greg. But what we're trying to do, this is an example of a genetics trial, and we're trying to delineate the individual trees and phenotype them so that we can understand, and actually phenotype them is cheaper actually at that level if you can get it working, automated. We've managed to do this with one trial, and actually it's about to come out in publication, and what we've found is that it works reasonably well with the growth traits. So height looks like it's really good. We can get a good heritability for that, and diameter as well. Based on the crown metrics, we can interpolate on the diameter, but some of the other traits like straightness and branching we've still got to work on, and I think that's the next step that we need to take. So in conclusion, basically, you know, I'm always a little bit cynical being a quantitative geneticist about genomics, and I apologise for that. But it actually gives us much more than I would ever have thought. And I'm really interested and excited about it now because it gives us quality control, gives us greater accuracy and ability to select our trees with confidence. And in turn, actually, that gives the industry more confidence as well. If you can show you've got greater accuracy, yeah, they're really happy about that. You can inform your breeding by understanding the structure of the populations, and you can hopefully, once we get our predictions working properly, then you know, we can start deploying these things straight away rather than waiting for 10 years for selection. And the key message here is genomic tools are getting cheaper. Like, I love that $20. We need to use them. We need to get them into the programs and starting to discover what we can do with them. It's already taking off in animal breeding, and they do it routinely. They do it routinely in crop breeding. So I know our genomes are bigger, but let's do it. Let's get on with it. And phenotyping and quantifying the impact of genetics across the forest is now possible. And this is pretty cool. And it kind of matches up with a lot of the ecological genetics conferences, talks that we heard yesterday even. And I do really believe that this is a way that we can inform our deployment and even our breeding, because if we can use some of that information to feed back about individual parents, then we may be able to augment what we already do. And remembering we're covering a lot more of an environmental gradient than we can ever do in a single experimental trial. So that's what excites me, really understanding the G by E. And phenotyping genetics trials and understanding how to get accurate measurements of an individual tree is what I'm now obsessed about. Cool. That's me.
Good. I didn't hear a ping, so that means we still have lots of question time.
That was bad timing.
Good planning, though. I don't know if you want to have feedback. Yes.
Nice talk, Heidi.
Thanks, Mike.
Have you been thinking much about phenotyping what we call the non-key traits, the sort of culling traits? Because it occurs to me that that's probably one of the easier hits you'll be able to make with that approach.
So by that you mean the wood, some of the wood quality, or?
Oh, not so much wood quality as stem quality, lead amalgamation, diplodia, deaths. Those are the sorts of things I was thinking.
Yeah. So what we're starting to try and look at disease.
Yeah.
So we've done a UAV scan of a trial for disease last week, I think. And in terms of the stem form, I think Dave Ponce worked a lot with the Zebedee, but we haven't quite worked through all the analysis of that to understand how accurate that is going to be, because we'll probably look at comparing it with Marvel, you know, the high density information about the form. So, you know, but that's next. I'm obsessed with that. I think that's going to be really important. Yeah.
Justin.
Can you tell a bit more about the way you're using the land surface models with the phenotyping? So I can imagine that environmental variation explains a huge amount, but it must be the residual, how big you are relative to where you're growing, that's the real phenotype?
Yeah, I mean, I think that's an excellent question. So actually the digital terrain maps are quite useful, and even that in itself is very interesting. But we've done an analysis on just a single trial again, comparing in ASREMOL the row column spatial effects versus, you know, a lot of spatial parameters. And actually the ASREMOL approach does really well, but the DTM, you can pull out certain sized areas at a certain focus, and it does add significantly to your accuracy. It just depends on how useful it's going to be and how practical it will be. But I think for microenvironmental analysis, it's going to be really neat. I really think DTMs are useful.
And is it possible then to use that at like intra-site variation at the E part to get a G by E? Like if you're growing really well, but you're down in a canyon with more water, maybe that's obvious. But if you're on a hill and you're like doing better than expected, then
that's exactly right. And in particular for disease, right? Because that's for us. We get Dothostroma in the valleys. We get red needle cast. Yeah, totally. And so for disease, yeah, that's really interesting. We are hoping to use, we've got fixed seed lots across these forests now, and so we're hoping to now fish them out and try and understand the environmental drivers of each of those seed lots. So there's lots more we can dig into with that data. Josky.
Joskin, then Jerry.
Heidi, to follow on from that question, so, you know, breaking apart the E and looking at the G by E back to genetics is really interesting, but I think, is there any thought of actually turning that around the other way and starting to work with the forest estate modelers and stuff and actually push this right out across the estate for estate planning?
Yes.
Et cetera, so.
Yes, absolutely. And Mike's nodding his head, and basically they're already using this for inventory. Yeah, so they already, that height surface they're using for inventory for their forest, and they use it for valuations and everything now. So you can break it up into coop sizes, and you know all the ages of that and everything, so they can track their performance and expected performance using LiDAR, and they do. It's cool.
Jerry?
Nice talk, David. Given the amount of acreage that's planted in eucalyptus now, or if you see it increasing in the future under global climate change, what do you think the proper effective population size is for supplying commercial seed?
I think in our context what we've got's okay, right? Because it's not a big deal, and we don't have... The only issue we have is probably that the Paropsis seems to fly over the Tasman every now and then, and we get a new one. So I noticed the other day there were 70, so we've only got about 4, so we've got to keep the diversity up a little bit for that, I think, and at least keep our breeding programs with a lot of diversity so we can go and fish in if we need to. But I think more clonal is okay if you manage it at the landscape level.
Good. We still have 1 or 2 minutes. Yes.
Yeah, following up on that, what is the genetic diversity of what you started with? However many years ago, was it collected geographically wide with that species, or I mean, you showed the genetic diversity compared to a seed lot from the tree seed center, but that was that multiple populations? What is that?
That's a really good question too. I think our diversity at the moment's a bit low, but we did start with, probably some. What's the other species of nitens now? Denticulata. Yeah, there's probably some denticulata that we're not recognising in there as a separate species because, well, it's all together. And but I think we've got a good collection in the base population. I think at the moment we'd be reasonably interested in trying to infuse a little bit more genetic diversity after 4 generations. And but I think it's okay for what we've got. It's only 13,000 hectares. It's not going to break the bank. But yeah, good question.
What was that being? Was it the 5-minute?
I really planned that badly, didn't I?
No, it's very good. I think it's an important discussion.
Yeah.
Do you have your hand up? Yes.
Greg.
Heidi, nice talk. I was really interested in the work you did on the looking at the stand level stuff, and the proportion of variation due to genetics was to me surprisingly high. What time period were you looking for those? I mean, if you're doing only stands last 10 years, why have they got crap genetics in them to give you that variance?
Yeah, I think that what's happened is that some of the new genetics has really come online in the last 10 years. So you've got a mix of like what they call GF19, which is kind of an average seed lot, and that's across the forest, and then they've got a lot of new clonal material. So I think that's part of it. But for them, it's showing the investment in clones is really working, and the investment in better genetics is really working. So that's probably why. Yeah.
Yes.
I was going to comment, Heidi, you made the point about the feedback loop, and when you're managing a radiata clonal program, it's really only a 3-year time cycle from getting that information and from the forest stands of clones and actually using it in the deployment program, so it really does make a big difference.
Yeah. And even if we can start working out at 3 years, feeding that directly back into breeding, because it is the operational performance of clones as well that we're really interested in, right? So we've got the clone, but now we've got to see that it makes a huge difference in the forest. So I think it could be really useful for that.
If I can ask a question, I mean, this is definitely a revolution in phenotyping. So do we need a matching revolution now again in genomics to keep up with the number of trees that can be phenotyped? And you said the frontier is a single tree measurement. So how do you think about that?
Yes.
What? The question is, what kind of? Do we really need to be able to genotype hundreds of thousands, potentially millions of trees?
Probably in this context, you won't need to genotype millions of trees because remember they're known seed lots.
And clones as well.
Yeah, and clones. So I think basically you can genotype your own deployment population, which might only be 30 genotypes. It could be 50, but that's really doable. And then you can even use that as a feedback loop into the genomic analysis even. I don't have a problem with, I think genomics is almost there. I think we still got a lot to do on the individual tree phenotyping actually. I think that's still the key. And even starting to work into functional genomics around what is the driver of that type of branching, or what is the driver of what's going wrong in certain areas, and feeding that back into breeding as well.
Good. Any more questions? In that case, let's thank Heidi for a fantastic talk. Good. So next up is Joskin Tibbetts. So Joskin is also a eucalypt enthusiast who's worked in many different areas of eucalyptus genomics. He's well known, probably being involved in producing some of the most extensive data sets for Eucalyptus globulus, and I believe he's going to be telling us today about how to leverage some of this information to do prediction of traits, quantitative traits.
Thanks, Xander. I'm going to reminisce just for 2 seconds because I actually came to this conference, well not this particular one, but its predecessor 20 years ago, and I was just about to start my PhD, and I'd done nothing at all. I'd come out of forestry, and I'm standing here, and I still feel like I've done nothing because there's actually just so much to do, and so it's actually really exciting. I mean, it's been like surfing a wave. I don't surf, but I can imagine what's happened in the last 20 years for me has been like surfing a wave. So I'm going to talk about, I mean, again, like Heidi, I've got a title I cut it down because it didn't fit. I want to talk about prediction and dissection of complex traits and focusing on Eucalyptus globulus, which is the main plantation eucalypt in Australia and around the world. So one of the drivers, I think, and there's a lot of other drivers, there's a lot of drivers Justin talked about in climate change, etc. But one of the other drivers is that we're, I think, shifting as humans, in fact, across the world, shifting from being basically gatherers of forest products, going out into wild forests and gathering forest products for building houses, etc., to becoming silvicultural. We're growing effectively crop plantations to produce wood needs. And I think it's really interesting. Victoria, or Australia, is, I think, at the forefront of that transition. So there's other countries in the world that still large. I think most of our wood products are still coming from native forests. But in Australia, we're now meeting about 80% of our supply from plantation forests, and that's a huge shift over a very short time period, and there's lots of reasons and drivers for that. But one of the things about the plantation forest is they're extremely productive for producing fibre and solid wood. And so off about just under 2 million hectares, we're producing 80%, which we were producing off some 100 million or so hectares of native forest. So I think that's a big driver. And so one of the questions is you know, we know breeding works. Traditional, I hate using the word traditional, but I can't think of another one. Quantitative breeding using phenotypes and BLUP and the advanced methods and statistical methods, which we know works. And one of the questions is, you know, can genomics help? And I think that question's been answered reasonably well in other fields, and so I'm going to confine it to, can it work in the Australian National Breeding Program, which is a rolling front program. So that's a breeding program architecture that's slightly different to other architectures where they do a bit of everything every year and you're trying to make gain and deploy gain all the time. And so Richard Kerr, who's here and is going to talk today, wrote a really nice gene level simulation model, and I'm going to sort of run through it reasonably quickly, but we basically simulated a breeding program that's like the globulus program, and started to try and ask questions of, well, you know, what happens if we do this and what happens if we do that? And so this is a base run. What you have here is this is the first trials, is going out into wild populations and establishing trials like we did, like Brad and others did around the place with provenances. And basically you wait for quite a period of time, 14 years, before the first phenotypes and reproductively available stuff comes through. So there's a bit of a flat period when you establish a breeding program where you're sitting around waiting, and people will do things like get the best provenances and etc. And then once you start breeding, the gain starts to kick off, and this is really what we're interested in, is this rate of gain, and that number there is just fictitious. So we actually we had to rescale it to make it a bit more similar to the breeding program. And so the index on the side is just the representation of value, and it's an index based on, in fact, it's a multi-trait index. It's based on the 3 breeding objective traits that are used in globulus. All the weightings are what's used in the program, etc. So we're really trying to look at what the program would be like. And so this is a base case. This is what we're calling a base case. It tries to mimic what happens in the program now. There's no genomics in there. 100% of the stems are measured for growth rate, for diameter, at age something or other, and about 20% of the trees are measured for wood properties, and that is about what's happening in the program. So that's density and pulp yield. One thing we asked straight up was, well, what if we just do a lot more phenotyping? Let's phenotype all the trees in the forest for all those wood property traits. And what you see is that the curve shifts up, so you do get a lift from doing that, and it's a pretty good lift, I think. I think it's an indication that that's actually a pretty smart thing to do, is do more phenotyping. But that gives you a sort of view of what range of gain can you get if you do actually do everything. And obviously there's a cost associated with that, which is why we don't do it at the moment, but it certainly does give you a lift, as you would expect. And so then we added genomics, and what we've done is we've said let's just do 20% of the trees with genomics, and the rest is all running off the phenotypes. What does that look like? So we're back to a case where we're doing 20% genomics, 20% still of the wood properties, and 100% of the growth. And what we see is that genomics actually kicks off it's the blue line. It kicks off earlier, and it runs up and eventually overtakes and is better than doing 100% measurements. And I think that's a really interesting finding. It says, yep, genomic information as an information source could probably add something to the program that's got value. So I don't think that's a great surprise.
Sorry, what age were you doing the genotyping?
At birth, yeah. And so what we see is that what the genomics is really doing, if you look at the generations and what their index value is, what we see is that the genomics, which is the blue again, is really pulling future generations forward. Yep, that's how it's getting this extra gain. It's pulling something that you wouldn't actually be selecting and making and crossing till down the track. It's bringing it to the now. And I sort of asked the question then, well, what may this mean for the forest? And if you look at just the top 1% of the three traits, and their index values are much higher, the genomics actually sort of takes off in the top 1% really much faster. And I think this is the sort of the business end of what goes on. So I think it looks like from our modelling, and I think from a lot of other modelling that's been done, and I think we're doing this to just give ourselves assurance that this could be valuable and to ask questions about what strategies we should use. It does look like we're going to get a good gain, and it's coming from this change in the generation interval that we're pulling them forward. And the breeding program would go from something like this to something like this as a model. And I think Heidi showed a similar sort of thing that they're thinking. And so we need to go out and collect DNA samples. And this is a cambial scrape. And so that's what we did. And so if you're going to do genomics, one of the things you need is a reference genome. And so for globulus, I've been bashing away at the globulus genome for years and years, and people say, why haven't you published it? And it's because I spend 99% of my time working on wheat and 1% of my time working on eucalypts. I wish it was the other proportion, but that's the way it is. So we started doing stuff on the globulus genome, in fact, to contribute into the genome paper, and some of this work went into the Grandis paper. And so that's actually how long it's been that I've been bashing away at it. And I sort of look at it as a good challenge to assemble a very difficult diploid. This is just an outcrossing thing. And the clone we have been using on is a Forest L. Menenko clone called X46. And Antanas is responsible for having captured that and spread it round, and they're now planted at the forestry school in Creswick and at the NGRC and here and there and everywhere. And so I can get samples off them pretty easily, or Antanas can, and he can ship them. And so there's 3 genomes, as we know, and I always put this in because this is Dot's chloroplast, and this, I think, represents a really big step in genomics, was just getting a chloroplast genome out, and a really high quality one. And in fact, that work has stood, I think, the test of time really well. And I love this picture. All right, let's go. So we've gone on. There's a picture of Antanas and who was that, Spocker? You remember? It was Ben Bradshaw. So Ben Bradshaw gave Antanas, and this is all X46 here, growing in clonal things. So we're trying to assemble all the 3 genomes. We've done a perfect finished chloroplast genome. We think it's got no errors at all, so it's 100% finished. And then we did lots of them. So Hossein, in his PhD, sampled from the Latrobe provenance progeny trials and did 20 or 30 trees from each provenance, and we assembled all those. And you can do really nice, beautifully resolved trees. And if you look at the distributions, this is nothing new. You can see chloroplasts are geographic, and they really cluster together. There's Tassie, but there are some of these funny chloroplasts from other places in there, and that actually represents the migration histories across the landscape, and they're known things. So, I think it's really lovely. And in fact, I know Brad's done this, but you can get down to locality with this. You could, from knowing a chloroplast, say pretty much down to a very close area where something's from. We've done the mitochondrial genome again to finished standard. I think it's not quite as high a base accuracy as the chloroplast. It's 411,000 bases, and this incorporated a long read type data, which was the Illumina synthetic long reads, which pretty much no one's really ever used. I used it as a beta testing program, and it produces really lovely long reads that are almost base perfect. So, but it's much trickier and harder than the current crop of long read sequences, the PacBio and the MinION. So it's not heavily used, but fantastic for assembling a mitochondrial genome. And yeah, the error rates in this are about 1 in 6,000. So not quite at the finished standard, which is 1 in 10,000 error rate, but pretty close. I'll just say as an aside, one of the interesting things about the mitochondrial genome is there's no evidence in globulus, we can't see any evidence in globulus that there's anything but a master circle of the way the mitochondrial genome's held. And that contrasts, in fact, to what we see in Grandis, which is that there are 2 parts to the mitochondrial genome. And that seems really stable within a species. So I think there's some quite interesting genome biology there. So the main genome, we've also got bionano data. Bionano data has been really useful in assembly of other things like wheat. It's again problematic to assemble into maps because of the heterozygosity. So there's no current map assembly algorithms that really do a great job of assembling bionano data into maps, and that's certainly something that I think is going to be worthwhile. But if I get to the finish, we're currently, I think we're beyond version 33. It's in about 13,500 pieces. Most of those pieces are megabase sized or half megabase sized, so they're quite big pieces. They're all at a very high standard of finishing. It is a collapsed genome, so it's not 2 haploid assemblies. And the size is very close. The total length of it is actually very close to the expected genome length. And so what I'm doing at the moment is getting some long-read MinION data, and also I've teamed up with someone to get high-C data, because it's really that high-level genome structure and ordering and orientation stuff that we're really missing. And so at the moment I still peg this assembly to the Grandis genome because it does have a pseudomolecule organisation, but I'd like to detach myself from that. So the other thing you need for genomics is you need to go out and get some samples and some sequences. And so one of the things we did was go out and collect from the breeding program, and Richard did what's called a Cholesky decomposition of the breeding program to identify all of the trees, the early generation trees or key ancestors that have contributed the most genes into the program. And you can see here there's some things that have contributed 6%, and then there's a long tail of things getting down to the bit of a percent, and I think that tail must continue on a little bit, but there's 83 founding mothers effectively underneath the globulus breeding program. And so we went on a mission to try and collect all of those, because I had a conversation with Ben Hayes about 8 years ago, and he said, got to get the key ancestors. I'm, killing myself to get the key ancestors in dairy, and they just don't exist anymore. And I said, the breeding program's got pretty much all of them. And he said, go and get them. And so we did. And so here's actually a pedigree of the program, and you can see here this is the ancestors, and there are a few that have contributed large amounts forward. This is first generation material, and second generation material, and then some of the third generation parents that are just coming online. And we've actually gone out and tried to collect that whole pedigree. There's not many of the Gen 0 trees, mothers that exist. We then had a second project where we tried to up the numbers, and we went out and we went to collect a few thousand what we called high value progeny, which are trees that are offspring of these, Gen 1 and Gen 2 trees, that have the most data. So Richard then went, rifled through the 300,000 trees and said, where are the ones with the most measurements? And we've gone after those. So there was a couple of big campaigns, and this is just one because it shows the commitment. Going out to trials where you're trying to collect just 5 trees, and the trial is defunct, so there's no markings, and it takes hours and hours and hours just to work out where you are. And a lot of the collections have been done by lots of people. We sent our PhD students, Wilson and Hossein and Fatima, out there, and I even sent my wife. And my now ex-wife. Thankfully, she's a forester, so it's okay. It was all going well until they got to Woolnorth, where there were leeches. And so I've taken all the trees that we've sampled and done whole genome sequencing on them all. In fact, Fatima's done most of that, and she's blazed new methods to make it tractable. And for the parents, I've sort of tried to get 10x coverage. Some of them are getting up above 30. And for the offspring, it's more skim sequencing at 1 to 2x. So they're not by any means sequenced to great depth, but still, I think this is a really good collection. And there's about 3,600 trees with some whole genome sequence from the breeding program in this set. Just a note on diversity to reinforce the picture yesterday. They're bloody diverse. So there's 8 SNPs within 100 base pairs here, and I think that's the point was made yesterday. It's like a horse and a sheep being the 2 alleles. And it actually has a huge impact on the bioinformatics. So most of the read mappers that are used have been developed for humans and work well in mammalian systems. And if you use settings and a lot of read mappers that can't go out, you might map in a picture that looks like this, which is really good. You've got some SNPs and these are different individuals. But in fact, the diversity picture looks much more like that in reality. So a note of caution, you have to be very careful to make sure you parameterise your read mapping properly, or else you get a lot of allelic dropout and a lot of misinformation coming through the genotyping process. So 2 minutes, crikey. I'll just skip that. I was hoping to get Heidi's time. I won't waste it. So one of the questions is what could we expect? This is just a graph of sort of looking at what sort of prediction accuracies we might have. And so at the point we are now, we might expect prediction accuracies for high heritability traits of about 50% and low heritability traits of maybe 20%. So we can't expect to be doing magic with the numbers we've got, but we should be doing reasonably well. So I've been using Bayes-R, which is a Bayesian model, and the nice thing about it is it allows us to look a bit at the genetic architecture. I'm not going to go into it. Basically, we're not fitting an A in this, which we could, and this is my colleague Ed, who's actually been rewriting the software on the fly so that I can incorporate the genotype uncertainty into the analysis, and this is generally how he looks when I turn up. So we ran models on some of the traits, the 3 breeding objective traits, and just a handful of the selection criteria traits. And the model fits are really good, but the question really is how are we going in validation? I did 2 by 10-fold cross-validation, dropping random trees. The fits look pretty good. There's some funny business going on, but overall it looks like in this model we're getting some real signal. in the data. And I'm not really interested here in the prediction side. I'm interested in what's going on under the hood with the SNPs and the variances. So we're sort of hitting about the place where we expect to be. And what we see is the architecture is allowed to vary here, and what we're seeing is that there's very large numbers. There's about 1% of SNPs are falling to things which have an effect predicted, and most of them are really tiny effects, which is sort of what we'd expect for quantitative traits. And these are the ones that we'd sort of pick up as QTL. And if you look at the variances, most of the variance that we could select on is in fact in this tiny effect bin. So it is a tale of saying we should keep selecting on all of the variants. And I'm nearly done. I just wanted to point towards the two posters, if you haven't seen them, from Wilson and Fatima on the Myrtle Rust project. So a lot of their work has fed into what I've done here, and they've got a whole lot of other stuff coming off on Myrtle Rust in there. So thank you.
Thank you very much. So, yeah, one question and then we have to move on.
Really quickly, I'm happy to see the genomic selection results having positive impacts over alternate feeds of selection. One thing I think we underestimate is that with molecular markers, you can assure the diversity outside the selected regions of the genome. So you can maximize the variability outside your target loci, and therefore reduce the risk of future unforeseen changes in climate and past pathogens. And I wanted to know or ask, have you tried to fold that into your evaluation? What is the value of genomic selection?
So I haven't done that. I absolutely agree with you. I think genomics offers an enormous array of potential other things we could do. The question I think we've been focused on is how do we actually operationalise it in a program without blowing the world up, and getting it to a point where you can actually move in a considered way and run simulations, models on real data sets to say what's the impact of that and should we do it. Because I think that's the decision making that needs to happen all the time. So, but absolutely I agree with you.
You base things on gain, and that's something you can convince your administrators of investing in. But there's a real value in reducing risk, and I don't know how to put a dollar on that.
Yeah.
That can be used to pay for the cost of genotype.
Yeah, I mean,
I think we have to, if you guys can do it over.
I used all my time. Thank you.
1% is gone.
Good. Next up is Jacqueline Gruber-Patinati. So you would know that Jacqueline was one of the early groundbreakers in understanding and studying lignin biosynthesis, and lately her work has moved to understanding also how wood properties and abiotic stress interacts, and she's going to tell us some of the interesting results that they have recently been producing. Jacqueline.
Thank you very much, Evander. And I would like first to this is the one to go the pointer. I would like first to thank our very kind and efficient organizers for setting up this very exciting meeting. And I'm also very happy to be here in the title. Title doesn't appear, but it was too long, like the others. So you've seen that I'm coming from France, and you can wonder, but is there any eucalyptus in France? If you see, we have a little bit less than in New Zealand, and all of the stories, the problem is, of course, frost resistance to increase the surface. And it was, I think, in the 70s, I guess, right, that Brad Potts and Bertrand Covin sent to France some seeds from Eucalyptus ghanii, the subspecies Divaretica, and as you heard yesterday from Rebecca. It's an endangered species, and I think tomorrow we should see some of the trees which are already dead. And I stole these words from a paper from Brad. For him, it's an iconic species for several reasons, and some are here. It's endemic to the Tasmanian Central Plateau. It's probably the most frost resistant among all eucalyptus species. It's also used and still used in breeding program in France as a mother in the crosses, and as I said, is a species which is really endangered, at least in Tasmania. And for me, it's also an iconic species because I've been, as Zander said, I've been working on lignin biosynthesis for a long time, and I think it's quite rare to point this, but for example, the first CCR gene, which is a limiting step in the lignin biosynthetic pathway, was cloned in this eucalyptus, not in Arabidopsis or any other model plants. And it's the same for some need master regulators that have been identified first in this in this species, and just the situation that we had even before the genome releasing the Higuandis released genome was that we identified in Guinea two new transcription factors. One was a positive promoter of the secondary cell wall deposition, and there was a balance with another one, which is a repressor of the secondary cell wall formation and lignin in particular. So we've been, you know, all that the activity of the vascular cambium and the differentiation into the wood and the secondary cell wall deposition is under a strong genetic determinism. For example, it's the case with juvenile and mature wood. So this is programmed. But it's also respond to external signal, environmental signal, and this has consequences on the structure of the xylem and also on the secondary cell wall, as for example, the well-known example is the tension wood, which is formed on the upper side of the lignin stem with this G layer, rich in polysaccharides, in cellulose in particular. Here is the case also if you play with nutrition. Here it's an essay in Brazil with nitrogen fertilization, and you see that if you have a lot of nitrogen, you get a very soft secondary cell wall in wood. And we've shown recently also that when you apply cold temperature, chilling temperature, we have thickening of the secondary cell wall very close to the cambium. So, in fact, we can consider wood as a biomarker of the interaction between the genotype and the environment. And when there is the tree is submitted to some environmental stress, like abiotic stresses, there is change in the regulation of the cambium activity and of the wood formation, and this occurs mainly through transcriptome modeling or other mechanisms like alternative splicing or epigenetic marks, and also metabolome remodeling, and this has consequence on the traits in wood. So there is less wood formed most of the time, and change in wood anatomy, and change in secondary cell wall composition. And this gives the plasticity of wood, and we want to know if these play a role in adaptation of trees to this abiotic stress. And also we are interested to know what are the consequences of these changes on the wood properties and wood production, because this is of course important with all the climate change we are that are going on at that time. Here is just a few insight in the work we've been doing after the genome, the release of the Eucalyptus brandis genome. We were interested to characterize some gene family containing members involved in secondary wood formation, and also, of course, genes of the lignin biosynthetic pathway. And I will not go through all of these. Just an example is the MIP family. So it's a family of transcription factor. And what we observed in this family, if you do comparative phylogeny with other woody species and model species like Arabidopsis or rice, you see that there are subgroups. Here are the genes. The family is more than 150 genes. And here you see this triangle are subgroups. And you see two colors, the yellow and kind of pink. You see that these are, in fact, subgroups of genes which are either more expanded in woody species or specific from woody species. Specific means, in this case, that they are not found in species like Arabidopsis or monocots like rice. And what is also interesting is that all these groups, either expanded or specific, contain tandem duplication. In fact, most of the tandem duplication occurring in this family are in these subgroups. Here is the strategy that we used to select candidate genes that we wanted to study, to characterize, and we used first this comparative phylogeny with other species and RNA-seq clustering. This was from Zander's group. And from this, we selected 200 candidate genes involving wood formation for fine profiling. And for this, we used 12 different tissues, and for abiotic stress, so mechanical nutrition, cold, and graft. And with this, we could draw some co-expression networks, so genes which are expressed together. And here you see that we found three main clusters. Of course, we are interested in xylem, and here you see in black genes encoding enzyme involved in secondary cell wall deposition. In blue, genes that we already know that are regulators of the secondary cell wall formation, and in yellow are new genes that have not been characterized before. And you see that here in the cambium cluster, there is a lot of new genes that have not been characterized in other species, or in particular a lot of transcription factor with no orthologs in Arabidopsis. So, and these we showed that some of these genes, a lot of them were responding. Here is to chilling temperature, but other can be activated by other stress. So to resume the criteria we used for to select these genes is expression in xylem, novelty, specific of woody plants or expanded in eucalyptus, co-expressed with lignin or non-secondary cell wall transcription factor, and responding to abiotic stress. And we made different type of construction and transform in different plants. We are able to transform eucalyptus, but it's so low efficient and so time consuming that we really did this for only one gene. and most of the other constructs would transform in Arabidopsis and poplar, but we were not so happy because it's heterologous system. So we decided to set up, homologous transformation system, which could be suitable to study secondary cell wall genes in eucalyptus. And this is the system. It's in fact transformation with Agrobacterium rhizogenus. And here you see what is a composite plant. The aerial part is not transformed, and it's only the roots that is transformed here in comparison to the control. And you can use a fluorescent protein to detect the transformed root. And here this is GFP and this is DS red. You can compare here, it's the same image, and you see that we have some roots that are not transformed and that you get off, and roots which are transformed. And as a proof of concept, we use my favorite gene, the CCR, because we know the phenotype. And you see here, comparing the wild type to the here was an antisense, that we have less lignin, and we have also vessel collapsing. And now we are growing these plants in hydroponic culture, so it makes easier the functional study. And we can also play with, we can apply some abiotic stress in the transformed plant or in non-transformed plant. And also recently, this is very preliminary result, we set up the CRISPR-Cas9 technology. So in this arrowroot formigradis, and we use again the editing of CCR as a proof of concept. And here you see the collapsed vessel in the transformed plants. So so we had deletion from 2 to 12 base pair. So, but this is our first success, but we need more confirmation and now to play on with other genes. So another message that I wanted to give you is that, in fact, in all our studies, of the new genes we characterize, a lot of them are giving, for example, drought tolerance or tolerance to some stress to the transgenic plants. So there is a link, a strong interplay between development, between secondary cell or xylem development and resistance to stress. So I think this idea is important for the future. When I started studying, the people were thinking there is gene for development, there are genes for which respond to stress, but it's not the truth. There is some genes which are hubs between the. I want just to give you a few insights in a system biology approach that we performed in collaboration with the CIRAD and with ESOLC in Brazil. It's using a E. grandis commercial clone, and it was a rainfall exclusion system. Here you see with 4 conditions, plus potassium, plus or minus water. And on this field trial, we did transcriptomics, metabolomics, wood anatomy and properties, and there was also some studies in ecophysiology. And to make long story short, here is a co-expression network of the genes differentially expressed between the different conditions. So you see when they are close, they are expressed together. And here in the middle, these are in the different color, are different modules of genes expressed in the same way in all different conditions. And module M1 to M6 are the one really related to secondary cell wall formation. And what is more interesting is that you can, in fact, you can integrate these expression modules to phenotypic traits. So you have correlation between some of the modules and some of the traits, like wood density, for example, vessel density or diameter, stem biomass increment, or lignin content we couldn't find, and saccharification yield or the other ecophysiological trait. And just an example, here Raphael extracted Raphael and Fabien worked on this project, extracted the genes which were correlated with some of the traits here, and they found some, we were interested by the regulators, so we found a lot of them which are already known in other species to be related to secondary cell wall formation, but we also found new candidates, and we used the error transgenic system to show that one of them was really correlated to giving to the phenotype consistent with the traits we were measuring, like here with saccharification yield and cell wall thickness in fibers. So this is to show you that there is possibility using gene network to highlight some correlations which are have a biological significance. And I would like to finish by giving you some overview of a European project we are participating. So no really results because it started mid of last year. So it's a project coordinated by Catherine Bastien from INRA Orléans. There is 19 European partners, 8 forest species, 6 native, 2 exotic. For us, eucalyptus is exotic, of course. And in fact, there are many objectives, but I selected 2 which are of interest. I think one is a genotype by site matching. One of the outcome of the project could be this one. And here is an example of what they've done in Sweden, the work of Mats Berling. So there is a kind of, they set up a kind of software where you enter the longitude and latitude of the place you want to plant your clone, and they will give you the list of the best from the worst clone to plant at this particular place. And the other challenge of this project is integrating biological explanatory factors into model approach to develop genome to try to improve genome-wide prediction. And this is driven by Leopoldo Sanchez in Orléans. And for eucalyptus, I'm finishing on this. Eucalyptus, we are 3 partners. So the CIRAD, the company Altri Florestal in Portugal, and our group. And so this is, we have some trial where we're going to do multi-level phenotyping, multi-traced, including drone imaging and monitoring of daily microfluctuation of the trunk and daily kinetics of climatic variables. Of course, genotyping using the chip, the 60K chip. This is everything is done on globulus clones, and we are also using a platform, a phenotypic platform for small plants to meet into drought and freezing conditions. And I'm finishing here with the acknowledgements, and I want to acknowledge all of you and show you some picture of Toulouse to give you that you are interested to visit. And
You're offering to host this meeting next day, is it? Thank you very much, Jacqueline. We're about 5 minutes late on the program, but is there any pressing question that anybody wants to ask right now? Okay, let's continue then straight with Carsten Kuhlheim. He's well known here in this community, formerly with Australian National University and now having moved to Michigan Tech University. And Carsten is going to talk to us about the genetic architecture of oil yield in eucalyptus. We look forward.
Thank you, Xander. Right, standing between you and coffee at the moment, so I'm glad to see you all here, and I'm very happy to present some of our results on genetic basis genomic architecture of oil yield in eucalypts. So most eucalypt species have a very large quantitative variation of essential oils in their leaf. That variation is typically 10 to 30-fold within a species. And you can see a population of one eucalypt species down here, with some individuals at almost zero oil in them, and some of them here, in this case 8% of dry matter oil. Some individuals have over 10% of essential oils in their leaves. I care about this variation and the essential oils because they confer ecological interactions with the environment, and also there are industries that are based on essential oil quantities. For example, eucalyptus oil industry and the tea tree oil industry. There are also thousands of chemical structures found in essential oils in eucalypts. These include monoterpenes such as alpha-pinene and 1,8-cineol, sesquiterpenes such as globulol. So within a plant, within a single individual, in a leaf, you can find 200 different terpenes in the essential oil. But most of the time, the profile is dominated by one or a few of these structures. As a geneticist, what I like about these guys is that they're highly heritable. So we find heritabilities of 0.3 to 0.8 for individual terpenes and compound traits of these terpenes. So that makes them quite good to work with. So if we look at this quantitative variation, you're all familiar with the classic genetic P model where you have two different phenotypes controlled by one gene. So here we have many phenotypes. We have a normal distribution. So this is affected by many genes. So how can we imagine many genes affecting one phenotype? So if you imagine we have a variation in a trait, in this case, say a terpene or monoterpene or a group of monoterpenes, we have a biosynthetic pathway. So we have genes in that biosynthetic pathway that can affect the variation in that trait. We also have a precursor pool, and in a carbon-based metabolite, the precursor pool or the precursor is typically produced through photosynthesis. And we heard 2 days ago from Antanas about variation in the large subunit of rubisco that changes efficiency in photosynthesis and can lead to variation in the amount of precursors that are available. So then we can have competing pathways, so we can have a different way this precursor metabolite can go. Then in the case of terpenes, which are toxic to the cells, we do need some sort of transport and storage. They can't be stored in the cells to these high amounts because they would kill the cells. This is always a balance between degradation, loss through volatile compounds leaving the plant, and biosynthesis. So these are all structural genes that are actually involved in biosynthesis of bits and pieces. But on top of this, we have a whole layer of gene expression regulation, protein regulation. So proteins can be degraded, proteins can be made. So we're basically going from a few genes in a biosynthetic pathway to a whole network which can include hundreds of different genes that could influence variation in a trait. So when I started working on terpenes about 11 years ago in eucalypts, we knew something about this biosynthetic pathway leading to terpene production. So that's where we started. So this is a schematic of the biosynthetic pathway of monoterpenes. We've got photosynthesis products, G3P and pyruvate, that go through the MEP pathway in the chloroplast. Precursors are produced in monoterpenes, and these can then further be modified downstream. So that's just a schematic overview. If we look at the individual enzymes that are involved in here, you don't have to look at all the details, but these are the different names, and we have the level of diversification where we get all the different terpenes produced at this level down here, the terpene syntases, which we also have heard about already on Monday, that they are very abundant in eucalypts. So 10 years ago when I started, there has been some work done in other species on these compounds and what the genetic basis of those were, looking at the pathways. So these are some QTLs that have been found, and they focus mainly on the entry steps of the MEP pathway, saying that this is really important. And then we have some down here at the bottom of MEP, precursor production and some terpene synthesis. So that was the stand of knowledge when I got into the field. So I'm first going to talk about a comparative study with using candidate genes, using that knowledge that existed 10 years ago. So we had 3 species of interest, tea tree, Melaleuca alternifolia, which is dominated by terpinane 4-role, Eucalyptus loxiflava, and Eucalyptus globulus, which are both, their chemical profile in the leaf is dominated by 1,8-cineol. So as you all know, Eucalyptus globulus is mostly grown for the production of timber and pulp, but some companies make use of the eucalyptus oil that is in the leaf and extract that as an additional way of making money. These 2 species, however, they are mostly grown for the production of essential oils. So they produce eucalyptus oil and tea tree oil are used for that production. So the study that we did here was we took between 400 and 500 individuals of each of these 3 species. We grew them in a common garden experiment. We quantified the terpenes in their leaf, and then we amplified 10 genes from the biosynthetic pathway that we knew had some effect, or that we hoped had some effect on terpenes. So what you will see in the next slide is basically we found genetic variants, single nucleotide polymorphisms, and we associated those with a variation in the terpenes. So the next table will show you basically these significant associations that we found. And it's sorted in the direction of the pathway. So here at the top we have DXS1, DXS2 and DXR, and down to the precursor production. So the first thing you can see here is that the 2 genes that people said are really important, DXS and DXR, there are no numbers up there, so we didn't find any significant associations within these. So these are sorted by species and then by type of SNP that we found. The second thing you can see here is that the majority of the genetic variants are found in introns, and they're not changing the amino acid composition. So we think that might have, or in the UTR, so this might have something to do with gene expression regulation. If we add up non-linked SNPs and look at how much of the variation can be explained, we go from 13 roughly % in Eucalyptus globulus to 35% in Eucalyptus loxiflava. So that is pretty good. We were quite happy with that, but we do want to find out more. I'm just going to give a quick example of this gene here, MCS. One of those 2 in Melaleuca alternifolia. These are the alleles found here at this position, a C or a T. The minor allele frequency is about 8% for the T, so it's a pretty rare variant. But here on the y-axis you can see the amount of oil. So the tea tree industry is really interested in enriching their population, their breeding population, for that T at that position. So this one SNP explains 9% of the variation of terpenes in tea tree, and is used currently as a marker in the Australian tea tree industry breeding program. I was looking back some time at a paper we published in 2013, and we found that the gene expression of that gene MCS actually correlated with the amount of terpenes found in the leaf. So what we have here is a situation where we know foliar terpenes are affected by MCS 1139. We know that MCS expression is affecting foliar terpenes. What we don't know yet is there a connection? Is this causative? So would like to go a bit further, but do need money for that, and we don't have that at the moment. Right, so this is just to remind me that we've been focusing a lot on oil concentration because that is a highly heritable trait, a trait easily measured, not influenced too much by the environment. But what farmers are interested in who are growing these plants for the production is oil yield. And oil yield is a function of oil concentration and biomass. So, for example, there is a tea tree plant that had a concentration of 24% oil per dry matter, but it only grew about this much in a year. So that's no good for anything. All right. So we could explain some part of the heritability. We're quite good with that. We looked at the biosynthetic pathway. We wanted to see what else is influencing variation in terpenes in trees. So for that, we did a study in Eucalyptus polybractea, which is exclusively planted for the production of eucalyptus oil. There's about 3,000 hectares planted in Australia, about 2/3 of that by 2 companies. These trees or bushes, their mallees, are harvested by coppicing every 12 to 24 months, depending on the amount of rainfall they got during that period. And then the oil is steam distilled from the leaves and can be either further refined or sold as raw eucalyptus oil. This is a picture, a Google map showing our progeny trial that we worked with. So you can see it's within this area here. Each family is planted in double rows along this side, along this axis. And just by looking at this, you can see that there's a fair bit of variation. We've got darker green areas. You can see some families here that are doing better than the families right next to them. So there's a fair bit of variation going on. So this trial is near West Wyalong in New South Wales. And here is a picture of how these balls of leaf look like when they're growing. Right, to the progeny trial itself. In 2009, about 83,000 trees from 40 open-pollinated half-sip families were planted there. Each of the mother tree is located within 50 kilometers of this trial site, so they're all relatively local. In 2011, we counted these trees, and so we see how many have survived seedling establishment, and thank you, Rose, for counting. And after the first coppicing, which happened in 2012, we counted the trees again to see how many survived the first coppicing. So then this is a little bit much work. Before we go into aerial monitoring or LiDAR, we have to actually do some work on individual trees. So we selected 480 trees. These were selected 12 trees per family. We then went and we measured their physical dimensions twice, height, diameter, leafiness, and we also sampled them for terpene quantification twice, same trees. We also took some samples and we extracted DNA, we did whole genome sequencing. We did a lot of bioinformatics then, that was a few years ago, so then the really interesting part came in, and if you're interested in what's going on there, please talk to me about it. All right, so then we had, after a lot of filtering, we got 2.4 million SNPs across the genome of Eucalyptus polybractea. The LD decay in the whole population is between 20 and 100 base pairs, so there's not much LD going on, which is why we're from the beginning reluctant to use a SNP chip or any other method. So then we associated the 2.4 million genetic variants with the traits of interest. Then we get a Manhattan plot like this here. So if you're not familiar with this, we have an x-axis here, which is the 11 chromosomes. Each dot represents a SNP that is associated with a trait that is shown here. And on the y-axis, you have a measure of a p-value, how strong association you have. You can see here there are 2 lines here. We had the upper line first when we submitted this manuscript, and all 3 reviewers were telling us we do need to do FDR corrections, so we did that, and now we're at the lower red line here instead. So we have more SNPs to look at. So when we look at what we find here, there's a lot of stuff going on in chromosome 1 with cinyl and alpha and beta pinene. This group of SNPs that associates with alpha pinene and 1,8 cinyl is in the proximity of 7 terpene syntases. And this one here with beta pinene is in the proximity of 1 terpene synthase. So we then functionally characterized these genes, so we cloned them, put them in E. coli, gave them precursors and looked what they're producing with the hypothesis that these are producing beta pinene and these cinyl and alpha pinene. So out of the 7 that we tried here, we got 3 genes amplified, and we got functionally characterized 2 of them, and this is one representative of those 2, and it produces 1,8 cinyl and alpha pinene, giving us a functional proof or showing that this is really what we expected. The beta pinene locus produces mostly beta pinene and a minor amount of alpha pinene. So then what happens when we look at the complex or the component traits where we add up all the different terpenes together, where we expected the biosynthetic pathway, the MEP pathway genes to be? Well, first of all, we didn't find them in the top 10 or top 20. They are there, but they're just not at the top of it. So what did we find instead? For sesquiterpenes, we found one really strong gene, which is a phosphoenol pyruvate translocator. That's a protein that is located in the chloroplast membrane, and it transports phosphoenol pyruvate from the cytosol into the chloroplast. Phosphoenol pyruvate is the precursor for sesquiterpenes, so basically it's taking away the precursor. For total terpenes and monoterpenes, we find a group of glycosyl transferases which has been shown to glycosylate terpenes, which makes them more soluble and easier to transport and store as well. Then we found this group of 3 genes, expansin, E3 ubiquitin ligase, and LRR kinase, and we found a paper that showed that not these 3 genes, but 3 genes of that type are required to form schizogenous extracellular spaces, which are similar to the oil glands in eucalypts. So while this is not very strong, we do want to go on and now test this. Are these 3 genes actually involved in forming the oil glands in eucalypts? An acetyltransferase, we think, is involved in proofing the inside of the oil glands so that the terpenes cannot escape from there. Right, so if we're going back to this, our hypothesis was here biosynthetic pathway influences most of the trait. These are our primary genes, if you want to. Apart from the individual terpenes, where that final step of the synthesis is really strongly associated, that wasn't the case. Instead, we had transport, storage for the monoterpenes and total terpenes, and competing pathways for the precursors. We didn't find many transcriptional regulators, transcription factors, but we can still look further into that. So I then go and go to Richard Davis, who runs this farm, and say, hey, Richard, we do understand the science now. And then Richard comes back to me and says, well, sorry, where's the money in this? He's interested in getting more yield. So the biggest SNPs outside of the sesquiterpenes explains about 3 or 4% of the variation. So unlike in Melaleuca alternifolia, we cannot use them easily as molecular markers in marker-assisted breeding. What we can do instead is we can look at a genomic prediction. So this is an ABLAB best linear unbiased prediction of oil yield in that population. And you can see while there is a correlation, it's not great. If we put in the G in the GBLUB, we get a much better prediction. So if we look at the prediction values, ABLUB for the different traits here, so this is height and growth, cinyl percentage, oil concentration. You can see it's almost doubling for many of these. For example, percentage cinyl, the prediction goes from 0.4 to 0.78, height from 0.08 to 0.16. So that is helpful in selecting individuals for breeding. And with that, I would like to thank the people involved in that. Hamish did the tea tree study. Amanda, who was spoken about on Monday about that mosaic tree, she also worked on the Eucalyptus loxiflava stuff. David here did all the Eucalyptus polybractea stuff. And then some of collaborators I mentioned here, including David Bush, who helped on the phenotyping side. Our collaborators from the University of Halle in Germany did the functional characterization. And thank you for your attention.
Thank you, Carsten. So, yes, Justin.
Carsten, on the prediction, how much yield increase did you predict? Not variance explained, but what is the kind of take home for the farmer? What was the increase in yield?
I would have to look that up. I'll get back to you tonight. I don't know at the moment.
Good. Any other questions? Yes.
Is there any kind of evidence for genetic correlations between the concentration of oil and biomass? Like, are they kind of intrinsically negatively correlated?
There have been, in eucalyptus, 4 or 5 published studies with contrasting results. Some show negative correlations, some show no correlations, some show positive correlations. We need larger numbers of individuals to really test this, and I think that there would be a plateau at some point of time, at some point of concentration. So like the example with tea tree that has 24% of the dry mass oil, it's a dwarf growth. If you are at the 10 to 14% range, anything below, it probably does not incur too much of a cost. So it really depends on your experimental design what kind of answer you get there.
Yes.
So genome-wide association studies can be biased against really rare variants. So do you have a feeling for whether there's the possibility that there's some genes that have multiple rare variants that may be contributing to the traits that you're not seeing?
Yeah, definitely. I mean, we did do some filtering of rare genes, so we had a cutoff of around 1% occurrence. We had a very moderate to small population here with only 480 individuals. We would really like to go a bit higher. They're definitely, and that's why you use models like a GBLUB where every single SNP is used in there, not filtered down by minor allele frequency. That way you really get the small contributions that we cannot get to be significant used in breeding.
Jasmine?
So, excellent talks, really interesting. The production system is a little bit like producing grass for dairies, and so it's a cut, sort of almost a sword system, cutting and regenerating. Is the plan with the LiDAR to really try and capture the productivity over the cycle of the plants and build that, because different plants will probably respond differently to this cutting cycle?
Yeah.
Through time.
I haven't thought about that much. What my idea or my plan is to use drones with the NIR to we can predict oil concentration and individual terpenes based on near-infrared spectra. Yeah. So we have these 59,000 trees, and we can only do 480 by hand. Yeah. If we fly over them, we can identify 500 or 1,000 trees that are, in the top percent. We can then go back and take leaf samples and test them in the lab. But yeah, as an identification method for superior trees, we would use mostly hyperspec. Yeah. Okay. And LiDAR for growth would be good to combine that all. Yeah. Yeah.
Good. Last question, Jerry.
I always get the last question for some reason.
You're sitting in the back.
You talked about yield and you looked at oil glands and growth and concentration. But what role do you think volatility plays in captured oil yields?
That's an excellent question. I've been working with this for a decade, and everything we do indicates that during the ontogeny of the leaf, the oil glands are formed and filled, and nothing else happens afterwards. They stay stable for 3 years, 4 years, 5 years with minimal loss. So they are really tight capsules until a herbivore bites into the leaf. That doesn't mean that there's no terpene production going on. Most of the terpenes produced in an adult leaf are volatilized and lost. So that's also why the phenotype we measured is not actually terpene production. The phenotype we measured is terpene storage during ontogeny of the leaf.
very good. I think we should stop here. Thank you very much, Carsten. Let's thank all of the speakers.
Hi, everyone. Come on in. You just had coffee. You're all awake. That's good. I'm coming from the University of Copenhagen in Denmark. And I just also like to start by thanking the organisers for their support, because I'm actually travelling with my 22-month-old that you may have met yesterday. But you may ask, why is someone from Denmark here in Tasmania presenting about eucalypts? Denmark is smaller than Tasmania, and they can't actually grow there. We have a couple in the botanical gardens, but they really struggle. And the plants in our glasshouse also aren't that happy. And while there is a market for Tasmanian princesses and their bridal bouquets in Denmark, that's really not the driver for eucalypt research in Denmark. But the research program that they have there really supports basic research, and it's heavily funded by private foundations. And fortunately for me, I'm funded by a private foundation known as the Willem Fonden, who really want to support interesting ideas, different species, basic science. And it didn't matter for them that I was coming as an Australian with my eucalypts. They thought and agreed with me that eucalypts are an interesting plant group to work on. So the work I'm doing, the title of the grant is How Does Climate Change Affect Plants and Their Herbivores? And I'm using eucalypts and koala as a model to understand this. But my project feeds into a much wider project with collaborators here in Australia out of the University of Western Sydney and UQ with Ben Moore and Philip Hugenholtz. And I would like to acknowledge my professor and mentor, Biwa Lindberg-Muller, who has kind of championed me and also given support via some money that we have through Big Centre grants. So my idea with this grant is to understand how eucalypts are able to respond to different environmental conditions. And as we know, eucalypts grow all across Australia in different climatic zones. And I would like to argue that their success lies in part to their ability to make many different specialised metabolites. We heard about the oils from Carsten just before. So we have these essential oils, monosesquiterpenes, but they make a plethora of other compounds. So many different phenolics. You can have, for example, phenolics conjugated to different terpenoid moieties. I've taken a big interest in cyanogenic glucosides, of which about 4% of eucalypts make. And these compounds are essential for how plants interact with their biotic and abiotic environment. Now, interestingly, we've done some imaging of eucalypt leaves. So in this case, we've done a leaf section, fired a laser down onto the leaf, and then all those compounds get flown up and get detected. And if we look into the oil glands, we get candidates that we would expect, so these FBC compounds, but we also get phenolics and we get many unknown compounds. And I think this is a really untapped and interesting resource that we can go into to figure out what compounds are actually present in eucalypts. And they may, for example, like these FPCs, have interesting pharmaceutical capabilities that we don't know yet. So my project broadly separates into some different themes. We've been working on some method development. We've been going and doing some metabolic profiling in the field and in glasshouse, kind of ontogenetic studies. We're interested in how these plants respond to abiotic stress. And then we want to understand the mechanisms behind this, so looking at pathway discovery and gene regulation to figure out what's going on. So interdisciplinary, multi-omics approach. But today I'm just going to talk a little bit up here, and then Cecilia, who's speaking after me, will talk about pathway discovery and regulation with cyanogenic glucosides. So in terms of methodology, we've been interested in these FPC compounds because they're known to be anti-koala feeding compounds. And in the past, they've been detected with LCMS and UV, but we've applied an MS method to detect them using characteristic fragmentation patterns, a bit like a fingerprint. So it means we can run our samples out, and then with the improvement of analytical instrumentation, we can actually see a lot more compounds than what we had previously. So previously maybe you'd see a dozen of these compounds, and now within a single extract we're seeing up to 49 different compounds. So when I talked about unknown compounds, many of these would be new compounds that we haven't identified before. So now that we have this method, we can look at different species and look at variation. And we can see, for example, in the leaves, some of the species produce really high levels of these compounds. Others are in trace amounts or non-detected. We get different amounts in the different tissues. So, for example, here we have Eucalyptus leucoxylon, and then there's not very much in the leaves, but if you look in the flowers, you have 40 times more of these compounds in the flowers. So that would have implications for reproductive biology of eucalypts. Again, we've looked at the imaging to see where are these compounds localized. For some of the compounds, we can see that they're right inside the oil gland, so stored within the lumen. But for others, they're actually associated to the oil glands, so inside, but also appear to be on the outside, and then even leading into the epidermis of the leaf. So they're sitting on the surface of the leaf. And that's interesting for me. In my mind, that could be a metabolic highway. So if you're having these volatile terpenes coming out, having these other compounds there may be a way of them being released from the leaf. We've looked at other tissues, so we can see these compounds in the stems and around the outside of the stems and oil glands. But also, this is a flower bud section, and we can see, for example, some of the FPCs here are in the surface of the operculum and the hypanthium. But other compounds are localized in the nectary and also in the pollen, so again, would have implications for reproductive biology. We're interested in, now that we have this method, interested in how these compounds respond to different abiotic stress. And this is with collaborators. For some species, this is a wounding ozone experiment. We see a response to wounding, but for other compounds, we see a response to ozone. For others, we see a decrease in FBC concentration. So they also would appear to be regulated in response to abiotic stress. So though these FPCs are non-volatile, they're being stored in the leaf, we know that they're involved in anti-koala feeding. And I was always intrigued that the koala will smell the leaf before it eats it. So what is actually being emitted from the leaf, and are those volatile compounds from eucalypts actually driving some of the koala feeding selection? And when I went into literature to figure out what is actually going on and what's being emitted from eucalypt leaves, very little has been done. In terms of flowers, nothing had been done. So we went out around Melbourne in an opportunistic study to see what was flowering, what could we capture. So we put bags around the branches and captured the scent from eucalypt leaves and eucalypt flowers. We took some different species based on whether or not they were making cyanogenic glucosides, koala preferred or not, and then depending on their flower colour and size and structure. Now, this was a very large data set. I don't have a lot of time, so I'll just give you a few of the quick take-home messages. The majority of the emissions was dominated by isoprene, but for me that's not so interesting. I'm interested in more the ecological and biological interactions that would be driven by these other compounds. So in total we saw 136 different compounds being emitted from the leaves. We saw the terpenoids, but we also saw a plethora of other compounds, so benzenoids, alkanes, alkenes, and the green leaf volatiles, which is the scent when you cut your grass. In terms of patterns of leaf volatiles, we didn't see any overall patterns between the different species. You can see there's a lot of variation between the species, but I didn't, for example, see a pattern of a koala preferred versus a non-koala preferred. There was some indication that defence strategy was important, so our most cyanogenic species, Cladocalix, had the lowest amount of terpenoids being emitted. But generally there was no overall pattern, so it would suggest that in a heterogeneous forest, all different eucalypt leaves are able to emit out a scent and they're able to communicate with each other. It's a different story, I'll skip that. It's a different story when you look at the flowers, and there we're seeing a very species-specific pattern. So if you go into the different species that we looked at, you can get a distinct profile between leaves and flowers. And when we plot all of that together, and so this is a PCA of all the different flower emissions, we can see that the species roughly separate from each other. And in addition to that, we seem to get a separation of the colourful bird-pollinated flowers compared to the white flowers. I'm rushing through this a bit faster than what I had expected. So overall with this, the take-home results from this is that I imagine that eucalypts in this heterogeneous forest, they're able to communicate with each other with the leaves. So a herbivore comes and attacks, and then all the trees in the forest would come and be able to respond to that. But in terms of flower pollination, they're really putting out a specific scent which would attract a particular pollinator. What was also interesting is that what is stored in the leaves and in the flowers is not what's being emitted. So, there's different regulation systems going on. We had a look at the diurnal regulation of these compounds, and there, I won't go into detail, but we saw something pretty interesting with isoprene being emitted at nighttime, which would not be expected. And then we're also interested in how these compounds are affected by environmental change. And the short answer with this is that yes, we do see a response, but I'm not presenting any of that today. So take-home messages is that we have new methods to be able to optimise, that we've optimised to identify and quantify these compounds. And I'm really interested in all these unknowns. That we have high emissions, and it would be really interesting now to look into species within populations. And finally, the environmental factors, we see that there is a response. And with that, thank the people involved in the project and my sister who's here taking care of my son so I can attend with you guys. In the field at the time, the student didn't see anything different, but I know in other studies there are many different pollinators coming at different times, and within, for example, our diurnal study, we're seeing different patterns, so some terpenoids coming up in the morning, other different compounds coming up in the evening. So it could be that you have bees or wasps coming at certain times and then moths coming at other times. But it really needs to be looked at in more detail. Yeah.
Rose?
That is really, really interesting. But when you're talking about the relationship between koala palatability, or palatability to koalas, and the leaf chemistry, was that done at the level of the species or at the level of individuals within species?
So in terms of the volatile emissions, it was, we had 6 replicates for each species, so it's not. So the more interesting biology will come from when you look at like one of the species that they're eating and then within species variation. And that's been shown for other compounds. So for example, within Verminalis, you'll have high and low producers of these FBCs, and they'll choose one or the other. What's interesting though is that of the species producing FBCs, they happen to be the species that were eaten by the koalas. So there seems to be some evolution going on between eucalypts and koalas.
All right, we're going to move on to the next half of your University of Copenhagen is Cecile Ketty Hansen. Characterization and evolution of cyanogenic glucoside biosynthesis.
Yes. So,
I'll just stand up. Yeah, I'll just stand up. Okay.
Is it too low?
I think it works.
Hello everyone. Is it too low or should I increase? Okay, good. Hi, my name is Cecilia. This is a PhD student from University of Copenhagen, and I'm going to present my project on characterization and evolution of cyanogenic glucoside biosynthesis in eucalyptus. So eucalyptus accumulates a great diversity of different specialized metabolites that are used to respond to and interact with the surrounding environment, for example, to attract pollinators or deter herbivores. So Lizzie already presented some of the diversity, so I don't need to go into detail with that. I want to add that some of the compound classes are present in all eucalypt species, whereas we have the FBCs and the cyanogenic glucosides that are only present in a few handful of eucalyptus species. As you know, eucalyptus contain many tandem duplicated genes compared to other plant species with sequenced genomes. If we look into some of the enzyme families often involved in specialized metabolism, then we also see that they have a high number of genes and also that there are many tandem duplicated genes. So Carsten and many others have already mentioned the terpene synthetases, but we see the same pattern for the UDP glucosyl transferases and cytochromes P450s. So the class of compound that I've been working on is the cyanogenic glucosides that you can see a few examples of here. They typically accumulate either as mono or diglucosides, and they're actually found in plant species distributed throughout the plant kingdom, such as sorghum, and we also find them in bitter almonds and even in ferns. These compounds are effective defense compound. They accumulate in the plant tissue, and upon herbivore or insect attack, compartmentalization of the tissue is disrupted, and the cyanogenic glucoside comes into contact with dissociation enzymes, and the outcome is release of toxic hydrogen cyanide. So we have studied the cyanogenic species Eucalyptus cladocalyx. Cladocalyx was actually the first eucalypt species identified to be cyanogenic. That was back in 1928, and in 1935 the cyanogen source was identified to be prunicin that you see the structure of here. And indeed we have found that prunicin accumulate in various tissues such as juvenile and adult leaves, in flower buds, flowers and fruits. And we have also identified a prunicin glucoside called amygdalin that we find especially in the flower buds and flowers. So in leaves, prunicin accumulation changes with plant age, and especially in very young plants we see a clear difference between different leaf types, with highest accumulation in the apical tips, then a decrease in expanding and further decrease in expanded leaves. So we set out to elucidate the biosynthetic genes for prunicin biosynthesis in eucalyptus. And the biosynthesis for cyanogenic glucosides is actually known in a few other plant species. And in general, it starts with an amino acid, which is converted into a corresponding oxime by a multifunctional cytochrome P450 from the 79 family. The oxime is next converted into a cyanohydrin by another multifunctional cytochrome P450 from the 71 family. And the cyanohydrin is finally glycosylated by a UDT85 to form the cyanogenic glucoside. And we thought it would be interesting to know the biosynthesis to further look into prunicin accumulation and the regulation of prunicin in eucalyptus. But based on this knowledge from other species, we initially envisioned that prunicin biosynthesis in eucalyptus would look like this. We would start with phenylaniline that would be converted by two multifunctional cytochromes P450, a CYP79 and a CYP71, and then finally have a glycosylation by a UDP glucosyl transferase UDT85. So we identify candidate genes and isolated from cDNA using a cloning strategy based on degenerate primers. And then we took these gene candidates and tested them by transient expression in the Nicotiana bentamiana. And in this experimental setup, we take our cloned gene construct, transform them, use them to transform Agrobacterium. Agrobacterium cultures carrying the genes of interest are mixed and then infiltrated into bentamiana leaves. Then we put the plants in the greenhouse, typically for 5 days or so, which allow them to express the genes. Then we harvest leaves and do a metabolite extract and analyze the extract by a suitable analytical instrument, in this case LCMS. So when we tested these gene candidates up here in tobacco, we did not see prunicin production as expected. But by combining these genes individually with already previously characterized genes from other plant species, we could confirm activity from the SUB79 and the UDT85. But the middle step was not working. So we went to look for a new candidate. And this time we obtained a transcriptome from the highly sonogenic apical tips. And here we see a bar plot of the most expressed cytochromes P450s in the tissue. We have our SUB71 and SUB79 candidate being the second and third most expressed. And then there's this SUB706, which is the most highly expressed in the tissue. So I thought it would be interesting to look a bit closer into this family. If we look at the number of SUB706 genes in grandis, there are 16, which is more than compared to Arabidopsis and Poplar. Only a very few enzymes that belong to this family have been characterized in other plant species, where they oxygenate either sesquiterpenes or flavonoids, so different type of compounds compared to the syngenic leukocyte, but still relatively small aromatic compounds. In the grandis genome, the SUB706 genes mainly localize into clusters with many tandem repeats. This might be the same case in Cladocalyx, and I thought it could be a good possibility for neofunctionalization of some of the members of this enzyme family. So I took the SUB706 candidate and tested it in our tobacco system, our benzimiana system, together with the other three candidates. And we see here, when we have all four gene candidates together, we get high accumulation of prunicin. Whereas if we combine only three of them together in various combinations, we do not get significant prunicin production. Okay, good. So now we had the enzymes that together could produce prunicin, but we had still not fully resolved the individual enzymatic step in the middle of the pathway. So in order to answer that question, we turn to in vitro assays. The cytochromes P450, they are membrane-bound enzyme, and in plants they localize in the endoplasmic reticulum. So I isolated microsomes from infiltrated benzimiana plants and administered the microsomes with carbon-14 labeled phenylaniline and analyzed the product by thin layer chromatography. And I will just jump to the conclusion from these two experiments. So in Cladocalyx, prunicin production looks like this. We start with phenylaniline, which is converted into phenylacetoalloxime by the SUB79. The oxime is converted into phenylacetonitride by the SUB706. Phenylacetonitride is hydroxylated to form mandalonitride by the SUB71. And finally, mandalonitride is glucosylated by the UDT85 to form prunicin. So here we actually had a biosynthetic orchestration that looked different from other plant species, where we have a multifunctional SUB71 that can convert the oxime into the cyanohydrin. So that was quite an interesting difference in the eucalyptus. So now we knew the biosynthesis in a single eucalypt species. But if we take a broader look at syngenic species in this genus, then we see that they do not localize in a single cluster. If we look at the phylogeny, they're actually distributed throughout the phylogeny. So we thought that prunicin biosynthesis would be an interesting case to investigate genetic signatures behind specialized metabolism and evolution. So I started to look into more species. I looked into 5 more species. You see here. If we take a closer look at the phylogeny of these species, then we have 4 that are cyanogenic, and then 2 that do not produce cyanogenic leukocyte. And I had transcriptomic data from 4 species and genomic data from 2 species. And if some of you are sitting on assembled genomes that I can look into, then please come and talk to me in the break. So with these resources in hand, I started out asking, 1, do the biosynthetic orchestration in other eucalypt species look similar to the one in Cladocalyx? And 2, do the non-cyanogenic species contain the biosynthetic genes and thus the capacity to produce cyanogenic leukocytes, or are there indications that they have had the biosynthetic genes? So this figure summarizes my findings. It's my last slide. So first of all, we found ortholog cytochrome P450 genes in the cyanogenic species, and we tested the activity from the one from Camphora and Euryanthes and confirmed the activity in prunicin production. So we see that the free subsystem in the eucalyptus is conserved, and in contrast to the 2 subsystem in other plant species. In the 2 non-cyanogenic species, I could find either full-length genes or pseudogenes for all the steps in the pathway. For the full-length SUB706, SUB71, and UDT85 grandis, we also tested them in the benzimiano system, and we found that they had the same activity as the orthologs in the cyanogenic species. You might wonder why there's no UDT85 up here. So when I looked at the transcriptomes from these two species, I did not find a UDT85 ortholog. And we actually had 3 time points for each of these 2 species where we also knew the prunicin accumulation. But using an approach similar to the identification of the SUB706, I found a UDT87 that could catalyze this last step in prunicin biosynthesis. So we have an interesting case of some pathway heterogeneities even within the same genus. Maybe Camphora and Euryanthes have had a UDT85 that were lost and then recruited a new UDT. There are still many interesting questions to look into. And with that, I would like to thank my 2 supervisors, Lisi and Biwa. Also thanks to a lot of members from the plant biochemistry laboratory and collaborators that have either been a part of this project or provided valuable inputs. And thank you for listening.
All right, we have 2 minutes for questions. Oh, come on. I'll ask you one. So, beautiful kind of examples of convergent evolution and gene family expansion and contraction. How much do you think regulatory variation and alternation of gene expression is important, as well as this gene copy variation?
Between species or within species, or
possibly within, or between?
Because we also see relatively big variance in the prunicin accumulation within the same species. So there could be different regulation of expression, yes, or genetic variation.
Cecilia and Liz, very nice talk. You addressed the presence and absence of GT85A among your species that you looked at. Do you have a feeling whether that particular enzyme is substrate specific, or is it a general as with respect to levels of transferase activity?
So I'm actually in the process of setting experiments up where I want to test the substrate specificities of these 2 glucosyl transferases. We know in general that the glucosyl transferases can be more promiscuous and take several substrates. So we're going to look into to see how specific they are towards the mandinonitride.
Let's go. Keep going now. We're going to take a little break from the biochemistry. We've got a tag team, Jules Freeman and Jacob Butler from Sion and the University of Tasmania, about QTLs for independent genetic control of myrtle rust resistance and native pathogens in eucalypts.
All right. Hopefully I can stand here and not stand in front of everyone.
I'll let you guys head off.
Okay. As Justin said, a bit of a change in pace now. So Jack and I today will be presenting some quantitative genetic and also QTL findings on our studies on host susceptibility to myrtle rust, and also comparing that to native pathogens. So we've been involved in some pretty big collaborative networks. The partners, or the funding bodies are listed below, and also all the authors on the main talks that we're going to present. And Simon seems to have been left off the slide, but shout out to them as my current employers. So, globalisation, as we've heard, it's really increasing the exposure of plants to pests and pathogens, exotic pests and pathogens worldwide. And myrtle rust really is a case in point. So from its origins in Central and South America, it's really spreading with increasing speed in recent years. too many countries to really list, but since about 2010 it was found in Australia, and it's been leapfrogging through Australasia and Pacific Islands, including New Zealand, where it hit in 2017. So myrtle rust really has a remarkably broad host range. A lot of rust pathogens, they might only affect one or a few hosts. Myrtle rust, the current host list globally is up around 500 at the moment, including, and that's all within the plant family Myrtaceae, and there's quite a lot of economically important species in that family that have been impacted. And notably, it's been a really big problem in the tropics, in Brazil particularly, in grandis and at hybrids in plantations. So when myrtle rust hit Australia, in view of this, the really big impacts it's had elsewhere, it really motivated a lot of research. Fortunately, the official line is it really hasn't had a really highly significant impact on eucalypts in Australia, either in plantations nor in native forests, but when it hit, we thought rather than wait and see what happened, we'd get in there and be proactive, and so a lot of really good research was initiated at that time by ourselves, and then guys like University of Melbourne, as you've heard, David Lee and Jeff Pegg up in Queensland, and also Carson's group's been involved in other taxa mostly. So just within the UTAS group in our collaborative network, we've looked at the genetic basis of variation. As you'll hear later on from Jack, we've done a broad screening within the Tasmanian eucalypts, actually screening all of the eucalypt taxa, and found some really interesting findings there. And we've compared resistance to native pathogens. And as you see, a recent publication, even in the post-genomics era, some pure quantitative genetics studies are relevant. So this is the study that I'll focus on now. So we focused on economically important species both in the temperate and the tropical regions, and key pathogens, key native pathogens of those species. So that was Teratosphaeria, formerly known as Mycosphaerella in Eucalyptus globulus, and Quambularia in Crimbia. And so we contrasted variation in susceptibility to these pathogens with variation in susceptibility to myrtle rust using common germplasms. Key questions we wanted to answer was, is there genetic variation in susceptibility? Because really when we initiated these studies, it wasn't well known, especially for globulus. Crimbia was a bit better characterised. Is there cross-resistance? So is there some relationship between the resistance to native and the introduced pathogens? And how is this resistance or variation in resistance distributed throughout the landscape? So we had pretty good sample size. In E. globulus, the samples were distributed across the native range. Within Crimbia, we had a better sample within one population with a really detailed sampling had been done there for Crimbia. So all of these families, we had an idea of the susceptibility to native pathogens. And for each of these families, we had 20 reps, so we're getting up into thousands of plants per species. So a key take-home message here, fortunately, the heritability indicates that there is significant genetic variation within populations, and that's for both the native and the introduced pathogens. The heritability estimates aren't directly comparable because we used inoculation in rust and so natural infections in the case of the native pathogens. Also, you'll notice there's a lot more differentiation across the landscape indicated by the QST for Mycosphaerella than there was for rust. So having this significant variation begs the question, how could this genetic variation susceptibility to an exotic pathogen arise? Generally, due to co-evolutionary dynamics, we expect that co-evolved pathogens, host species there'll be variation in susceptibility, genetic variation in susceptibility to co-evolved pathogens, but that's not really always expected for naive hosts when these new diseases come in. So some candidate explanations are, it might be pre-adaptation, there might be some sort of constitutive differences, say leaf chemistry or secondary metabolites that have been selected for by other forces, which are also conferring variation in susceptibility to these new encountered pathogens. Might be some induced mechanism, or there's been a suggestion there's ancient R genes floating around from a past exposure of the eucalypt ancestors to a rust pathogen. So basically our findings are suggesting that pathogen general mechanisms are unlikely. So there's no quantitative genetic correlation within populations in globulus to the Teratosphaeria versus myrtle rust. And as Jack will show you, the QTL evidence argues as well that it's generally independent in these within host species at least. This one gets a bit busy. The take-home message is, so we know there's significant genetic variation within populations, but when we look at the distribution of this variation for myrtle rust, basically there's no real clear pattern across the landscape, which is a contrast to what we see in terms of the native pathogen, and that's reflected in the QST. In the case of globulus, it was really no more than we expect by drift alone, which we see by comparing with FST from neutral markers. There was no real difference. In fact, FST from markers was higher. Again, it's not correlated with globulus. And a really interesting finding was we actually found, in the case of the native pathogen, in populations that were more susceptible, climatically more susceptible to Teratosphaeria, they're actually more resistant, which argues that pathogen-imposed selection has influenced the pattern across the landscape, perhaps. In terms of myrtle rust, we actually found the opposite. So in drier populations, which are likely to be less susceptible to fungal pathogens, they're actually more resistant, which provides some so I suppose a potential explanation, or it points to that maybe there's something else that's been selected for in these drier environments, and that's broadly consistent with the findings from the University of Melbourne, where they're finding evidence for pre-adaptation influencing myrtle rust. Okay, I'll hand over to Jack while he's still got time, and he'll do some general conclusions at the end.
Alrighty.
Talk much.
So, part of my PhD was studying QTL for rust resistance and for native pathogens in both Eucalyptus and Corymbia, and contrasting the two to see if we can support some of the findings that Jules was talking about. So, if you're looking at a QTL, you basically need a linkage map, and we're looking to associate the phenotype in our mapping cross with specific markers on the linkage map, and then we can sort of identify an area of the genome where we expect that genes or other things influencing that trait will be located. For rust specifically, there was a lot of work done earlier on in the 2000s where they found a major QTL for rust resistance in Eucalyptus grandis. They called it PPR1, Puccinia pseudo resistance 1. And that was part of a large breeding effort. But it turned out that in 2012 they found that some of their resistance populations were starting to get knocked out by rust again, suggesting that that single QTL isn't the only thing controlling rust resistance and that it can sort of overcome it. So we got into looking at it in Eucalyptus globulus and in Corymbia. So we needed two linkage maps to do two QTL studies. This linkage map was created in globulus by Corey Hudson in 2014. Unfortunately, we did for the Mycosphaerella, or Teratosphaerella, sorry, resistance was all based on field data, but for rust, we needed to use artificial inoculations, and you can't do that on adult trees because rust likes to target growing shoots and apical tips and all that kind of stuff. So what we did is we studied 15 open-pollinated progeny, the F3, from these F2 trees. So we ended up screening about 3,200 trees in this cross. And then in Corymbia, this cross is a little bit funky. We had two mother trees and a single father tree to create two separate crosses. So we had a bit of a technical replication in the middle. I'm going to have to speed up. So this came us with 4 different linkage maps we could use to look for different QTL. So in Eucalyptus globulus, we found 2 QTL for the immune response and 2 QTL for the hypersensitive response. We score rust resistance on a 1 to 5 scale, immune, a hypersensitive reaction, and then various scales of infection. And we thought that there might be something in that there's sort of a separation between the hypersensitive and just being flat out immune. So we decided to score it in this way. So yeah, we found 4 QTL, and there was no overlap with PPR1, which was fairly interesting. We went to Corymbia. This one's a little bit busy because we've included some of the other native pathogens, but we're just looking at these QTL here across our 4 linkage maps. Again, we found 3 QTL for the immune response and 3 for the hypersensitive response. What we found is that when we place all these QTL from various different eucalypts onto the same genome, we find that the majority of them are independent. They're not finding a common QTL that overlaps. Apart from these 2, one from Eucalyptus globulus and one from Corymbia on chromosome 3. And that's interesting because chromosome 3 is one of the more conserved ones, but I can't get into that right now. But when we start throwing in the rest of the QTL for native pathogens and other things in Eucalyptus globulus and Corymbia, say QSB, Mycosevilla, Asondarunia type 1, we find that most of them are fairly independent, but we're starting to see some sort of co-locations, which suggests that there's things that might be in common between these different pathways. There's all kinds of things we could explore in there, but I'm not going to have time to. So in summary, there's evidence for just strong genetic control and variability and susceptibility to myrtle rust in the eucalyptus examined, which is good to see. There's diversity between species and within and between populations. And since we're not seeing all these things stack up in one place, it's likely to be multigenic, involve multiple different mechanisms. We're seeing that susceptibility to myrtle rust isn't correlated with the native pathogens, so suggesting that there's separate mechanisms or separate resistance responses influencing that. And it's good to do this sort of thing because it's interesting. Thanks.
You guys can both come up. We got 3 minutes for questions.
Oh, I did this.
Sorry, Jack. I realised we're right off the microphone.
That's all right.
I'm curious about the correlation, and I think you presented data that suggested you would be able to show the correlation between the field and the glasshouse inoculation results.
Yes, I didn't present it right there, but yeah, certainly in the study we found with Quambularia we were able to do an artificial inoculation and also compare that with field results, and the correlation was really good. Basically across, we had the source studies for Corymbia had Quambularia infection across a whole lot of different trials, so it's highly likely there are all sorts of different strains of the pathogen, and regardless of that, the repeatability, the correlations were really good.
I was specifically wondering about myrtle rust, though. Did you show variabilities of field and...
Yeah. Sorry, there's we did not have we haven't scored myrtle rust in field trials. It's because, as I mentioned earlier, there hasn't been a high level of infection in field trials. So yeah, if we had a situation where you could potentially do it, but it's just too patchy, the incidence of myrtle rust in field trials. So we haven't been able to assess that.
Jerry and then Paul.
This is a basidiomycete, I assume. And what spore form causes the disease, and what's the alternate host?
Teleospores in the myrtle rust, I believe, and there's some debate about that because they haven't been able to find. There's only a couple of papers now, scant evidence for myrtle rust completing its life cycle. So very little evidence of sexual reproduction. And there has been a hypothesis that that's because it's actually completing its life cycle on an alternate host, but as I understand, they haven't been able to verify that.
I just wanted to pick up on the comment that there wasn't any association with the native pathogens, but maybe the dry environments, and whether there's any particular attributes or traits back to the QTLs, whether we know those QTLs, while you didn't map it specifically here, there might be other traits that we have mapped in the past. Is there any sort of comments about which traits or underlying genes might be important?
Since we've got the Eucalyptus grandis genome and now the fresh Corymbia one, we can go looking for candidate genes under these, positional candidate genes under these QTL, and we're finding a lot of nucleotide binding leucine rich repeats, the NBLRRs. We're also finding associations with microRNA precursors, which are associated with all kinds of things, and so they could be helping to influence this. We have to pay attention to the fact that a QTL is scored in just a single family, so we might not be seeing the segregation in that particular family that's found out in the wild or in other things. So this might not be all there is, effectively. Yeah.
All right, we better move on. I think we have a very short couple of talks now. 5 minutes for Julia Candotti from the University of Victoria. Genetic dissection of growth and wood development in nested, multi-parent eucalyptus hybrid population.
Okay, good afternoon everyone. So today I'm going to be giving a brief overview of my MSc, which is the first steps towards being able to dissect growth and wood development within a multi-parent mapping population of eucalyptus hybrids. And this was done under the supervision of Professor Nyberg. So multi-parent populations are advantageous as they combine the high power of linkage analysis with the high resolution of genome-wide association studies. And these populations have been used in a number of crop species. So shown here are two examples in maize and tomato, and both these studies showed how these populations can identify a large number of QTLs of varying effect across all families. So this just further confirms that these populations have a high power to detect QTLs combined with the high resolution of genome-wide association studies. So in South Africa, we work very closely with SAPI, which is one of the forestry companies, and they have performed a number of interspecific crosses between Eucalyptus grandis and Eucalyptus europhylla. They have then taken the F1 progeny and clonally propagated them, and we can capitalize on these crosses to identify markers for traits of interest as well as for genomic selection. So using these crosses, we constructed a Eucalyptus multi-parent population by crossing 9 Eucalyptus grandis pollen parents with 8 Eucalyptus europhylla seed parents. But today we'll just be focusing on a single half subfamily, and the numbers inside the trees represent the number of seedlings that there were for each of the crosses. So we confirmed parentage and species identification and sent the samples for SNP genotyping using the Eucalyptus 60K SNP chip. And of the 60,000 markers assayed, 2,143 were pollen parent informative, which means they were heterozygous in the pollen parent and the same homozygous class across all seed parents. And this is because we could then trace the segregation of the pollen alleles throughout the progeny. So using these markers, we first constructed high-density genetic linkage maps, and shown here is an example for chromosome 8. And then using these maps, we constructed framework genetic linkage maps. So our criteria for these maps were that no two markers should be closer than 1 centimorgan or further apart than 10 centimorgans. So these are the 11 linkage groups for the framework genetic linkage maps, and we had a total of 391 markers with an average marker density of 2.5 centimorgans. And in the near future, we will be mapping QTLs controlling growth and wood properties onto these maps. We then analyzed the segregation distortion patterns of these mapped markers. So our pollen parent was always heterozygous, save for alleles A and G, and what we expect is that there will be an equal proportion of alleles A and G within the progeny. However, sometimes the one allele will be favored or the alternative allele will be favored. And we can visualize this in this graph. So this is a graph for one of the full subfamilies, and on the x-axis is the markers per chromosome, and on the y-axis is the amount of segregation distortion. So if a marker has a value of 0, it means that both alleles were present in equal frequency and there was no distortion. If it is above 0, it means the one allele is favored, and if it's below 0, it means the alternative allele is favored. So when we add all of our full subfamilies within our half subfamily, we see that there's a large amount of segregation distortion across all chromosomes and across all of the families. And what is interesting to note is that in some cases, one allele is favored in the one family, but the alternative allele is favored in the other family. So all of this segregation distortion is starting to suggest that there are multi-genic mechanisms underlying hybrid compatibility. And if we go back to our multi-parent population, we see that a large number of the crosses did not yield any offspring, which suggests that those parents have incompatible genotypes. What we also see is that some of the parents have a generally poor combining ability in that they do not combine well with any of the other parents we have, while other parents have a very specific combining ability in that they combine very poorly with some parents but produce a large number of progeny with the other parents. So what we would like to start to do is to resolve the haplotypes underlying the incompatibilities using long-read sequencing. So with that, I would just like to thank my supervisors, the FMG research group, and funding bodies.
Better jump straight to the next one, but I think you're getting at the speciation genes there. Larissa Macchiato-Tobias from the University of Melbourne, tubulin genes, tubulin roles in determining cellulose microfibril angle during wood formation in eucalypts and other forest trees.
Well, hi everyone. So I'm Larissa. I'm from the Forestry Biotechnology Group at the University of Melbourne, and today I'm going to talk a little bit about my master's project. So first of all, what is cellulose microfibril angle, or MFA? So MFA is measured as the microfibril deviation from the long axis of the cell. And this feature is especially what we can measure in the secondary S2 layer of the secondary cell wall can be determined for tracheids and fibrous qualities, such as stiffness or elasticity. So, in cells with more longitudinally aligned microfibers and consequently low MFA, they can resist to high tensions, whereas fibers with large MFA provide more elasticity to wood and can resist to damage due to wind forces. And we already have an idea of how the cellulose microfiber pattern is determined. It has an association with the microtubule cytoskeleton, and in 2006, this study here by Paredes and colleagues demonstrated that microtubules share spatial temporal location with the cellulose synthasis in the plasma membrane, and they move along tracks that are delineated by microtubules. This is for the primary cell wall. And in 2015, this other group, Watanabe and colleagues, demonstrated the same mechanism for secondary cell wall. And other studies are working on identify the linkers between microtubules and the cellulose synthase complex. Okay, MFA plays an important role in what is called reaction wood. The wood forms a response to gravitational stress. So, for example, in angiosperms tension wood, we have really low MFA values, and that kind of acts pulling a leaning branch upright. While in gymnosperms compression wood, the large angle which microfibers are deposited creates a compressive strain that pushes the leaning branch upright. And it was demonstrated that tubulin genes, which encode the primary components of microtubules, are highly expressed in reaction wood. And were also demonstrated to affect the MFA of transgenic xylem fibers in eucalypts. So tubulins. So let's say we have an angiosperms branch. We have tension wood being formed in the upper side of the branch, the cells with really low MFA, and tubulin genes being really high expressed. So presumably we have more tubulins, more proteins there, and we don't know what they are doing. We don't know why they need to be in large quantity in this kind of wood. So our hypothesis is that this differential expression in tubulins leads to a differential microtubule assembly. So it led us to some questions that we possibly will answer in the next few months. So how microtubules are organized or orientated in reaction wood when compared to the wood formed in the opposite side and the wood forming stems growing upright. How microtubules are organized and orientated in tension woods of angiosperms when comparison to compression wood of gymnosperms, and what will be the effects of a disruption in this microtubule array for MFA determination. So basically the objectives of my work is to determine the microtubule organization using immunolabeling techniques from three species: Eucalyptus globulus, Populus alba, and Pinus radiata. So we expect to see something like this. These are longitudinal sections of eucalypt fibers, and you can see a difference in microtubule organization in tension wood to opposite wood. So we are trying to correlate microtubule organization to specific MFA values. And separately, we're going to use a microtubule inhibitor in cells from tissue culture of the same species to determine what are the effects of a microtubule disruption in MFA. So with this, I expect to shed light on how tubulins participate in MFA determination in secondary cell wall of xylem cells of woody trees. And that's all I had. I'd like to thank the supervisors, lab mates, the Gen organization, and thank you all.